Hello everyone, and welcome to the Genomics show. I'm your host, Maria Nadestad. Today I'm going to be talking about some of the trends in genomics that I am most excited about. And this was actually a little poll that I did on my YouTube channel here, and this was the top option that people chose. So that is the one we shall commence with now. So I initially wrote down a list of all the things I'm most excited about. But then I thought, okay, let me try to organize it a little bit more so that we can see how they all fit together into the larger genomics field, and I'm not just like listing cool things, because this isn't a listicle. We are trying to build a shared understanding of what is going on in the field, and I'm not going to be able to cover everything, but I will let you know where the gaps are and where there's more cool stuff that you might want to check out. So the way that I think about this as I was making my list is I first put down the Telomere-to-telomere project and pangenomes. These are both something that's really affecting my area of the field, which is where we just focus on reading the genome very accurately. And telomere-to-telomere is essentially finishing a whole human genome for the first time. This is new because the original Human Genome Project did not actually fully finish the human genome. And in telomere-to-telomere, they were able to actually fill all the gaps that were originally there and have been there in every other iteration of reference genomes that we have tried to make as representations of the human genome. They have all had gaps because it's a very hard problem to actually fill all the gaps due to repetitive elements, places that are harder to sequence, and just, lack of some of the technologies that we have today, like long-read sequencing. So this just finished telomere-to-telomere, and you should definitely check it out if you haven't already. If you work in genomics at all, you should be aware of the T2T project and try to see if it's possible for you to use this new reference genome in your own work. It'll be interesting to see if we can get the whole field to move on to this new reference genome, because we moved to GRCH 38 officially. It came out in 2013, which is right about when I joined the field. And some people are still using HG19, which is also known as GRCH 37, so it comes right before GRCH 38. And there's some people who still haven't moved on to GRCH 38, so I'll be really interested to see if we can get people to move on to T2T entirely. Now, the other one that I'm excited about is pangenomes. And these things go together a little bit because the T2T reference and this project is also creating additional full telomere-to-telomere human genomes. The first one was haploid, which is CHM13. That is slightly easier of a job. It was still a lot of effort for the team, but it is easier to finish a haploid genome because you don't have to worry about phasing the two haplotypes apart. Whereas with normal diploid genomes, it's somewhat more of a challenge. So they're working on doing more diploid genomes, and then those will fit into the pangenome. What is a pangenome? It's, if you know the Greek pan, it's like all. So the idea is that you have essentially a set of many genomes in some kind of data structure that lets you consult them all at once as a set, instead of only looking at one genome at a time. You are kind of looking at all of them. And the way that we usually visualize this and think about using it is in some kind of a graph format, where you would have regions that are very, very similar as one node, and then you have a place where there's more variation, where you break out into multiple pieces, multiple different haplotypes. And then maybe you come together again when it's simple again. And that's kind of an oversimplified toy graph I just drew with my hands there. It gets a lot worse than that. It gets a lot more complicated. But this kind of thing can really help as we are sequencing and analyzing a new sample, because the sample is not going to fit best to any one genome that we've already sequenced. It will be probably fitting with a combination of haplotypes from different genomes that might make up our pangenome already. And figuring out, oh, it's really close to this one, but with these variants, can be a lot more helpful than, oh, it's got all these giant structural variants relative to the reference genome. But it might be that we've seen that exact set of structural variants before. For one thing, the reference genome might not even have the particular sequence that you are seeing in this new sample. But if we include many genomes and make a pangenome reference, then you can still map them to one of the haplotypes, and you might be able to actually use that sequence now, which would normally be thrown away entirely. And so there are things that we can do to make use of that. Now, you might be thinking, oh, but how am I going to make use of the sequence that's mapping to a pangenome somewhere that we don't have any previous information about other than it's been seen in these other samples? So that's where you kind of have to build on top of this framework, and all of the studies that we have done relative to the old reference genomes, we kind of have to also try to transfer some of that information into the pangenome space, and we have to do new studies. We have to figure out how to make a new collection of information for these regions that we weren't able to access before. And that's kind of an exciting challenge because the whole field has to, continue to build the knowledge on top of that. So I think, telomere-to-telomere and pangenomes both go into reading the genome accurately. And the other major thing that goes in there is just using long-read sequencing itself. So PacBio sequencing and Oxford Nanopore sequencing have both really come into their own in recent years and have also both contributed to the advances that we've seen with both T to T and starting to take shape with pangenomes. But they're also contributing to overall being able to read every new sample more accurately, whether we're making a reference out of it or not. So I think of reading the genome accurately as our foundational block that the rest of the genomics field is built on top of. So this is the part of the field that I have been most focused on in my career. I am not necessarily part of all of these efforts for sure, but my work builds on top of it. And I know many of the people who have done some of this foundational work, and I have certainly used a lot of long-read sequencing in my career so far. I would say that's most of what I have been doing is using long-read sequencing and trying to make sense of the results, trying to make some visualization tools that help us see the context we're getting out of that more clearly. So I have been focused in this foundational block. So that is the area that I will know the most about. I do want to talk a little bit about the things that build on top of it as well, though. So the second block, I would say, is finding connections between genotypes and phenotypes. So now that we have read the genome accurately, it is time to draw connections between those ATCs and Gs and actually saying like, oh, if they have an A here, they are taller. That's a really dumb example, but you get the idea. So it's the genotype to phenotype connection. So this can be done in many ways. One of the exciting things that's happening right now is that we have access to larger data sets of actual humans. So something like the UK Biobank, the All of Us project, which is similar, but in the United States. So you also have a more diverse population there for sure. And there's other large data sets like this. And these are enabling us to get phenotypes from a large diverse population, as well as genotypes. We are definitely, trying to do sequencing, at least exome, followed by whole genome, maybe also some long read sequencing. I think all of these projects are planned to have a combination of these, but it's kind of, they're stepping up over time how much genomic data they're able to get. But the phenotype collection is a really big deal as well. And basically just having a bunch of genotypes for a bunch of people is not super helpful without the phenotypes. Because unless you're measuring a phenotype, you can't draw a connection between that and the genotypes. So it's starting to be that the phenotyping is actually also very much a bottleneck for drawing these connections. So the large data sets are doing a lot of phenotyping. They're doing a lot of genomics as well, and that's very exciting. This enables us to do large GWAS studies. So this is genome-wide association studies. If you haven't heard of this, you should look it up. Definitely be aware of what that is. But it's very exciting. It's basically where you, at a large statistical level, draw connections between the genotypes and phenotypes of a large sample of people. Another thing that you can do to draw connections between genotypes and phenotypes is molecular biology, right? You're investigating the mechanisms whereby a genotype affects a phenotype. So there's a lot of molecular biology in here as well. And some of it does depend on the genomics, right? In the olden days of the field, when some graduate student could spend their entire PhD thesis on just getting the sequence of a single gene, that was, partly to help understand, if you can understand the sequence of the gene, then you can figure out the amino acids, you can get an idea of what's going on for the protein, and you can also, start to think about, what happens if I'm seeing different mutations in it, like different variations in this gene, how is that affecting the phenotype I'm seeing? And now we can do that at a much larger scale because the genotyping has become easy with sequencing. But that still means that, doing molecular biology at a scale where you can collect phenotypes automatically as well is super interesting. I've seen cool things, for instance, at a conference where they're phenotyping plants using cameras and AI to just automatically measure things about the plants just by having a camera zoom over a whole row of plants in a greenhouse. So there's a lot of interesting things you can do with large scale phenotyping. And yeah, like I said, all of molecular biology also kind of fits in here, finding the connections between genotypes and phenotypes. So the third block that goes on top of this is, now that we have the connections, we want to use them to inform patient care. We want to use them to make predictions. So because of all the work that has been done linking genotypes to phenotypes, it has now become more worthwhile to sequence patients to try to understand what is causing the phenotype that you're seeing in the clinic. You can try to get an idea of, what gene has changed. Some of this goes back to the molecular biology. Somebody has manipulated these genes before in another organism and has an idea of which biological systems that might be impacting. Look up ontology if you want to know more about that. And then you also have all the GWAS results, right? Finding these statistical connections. And all of that means that sequencing patients, or even just genotyping them, with SNP chips, this is the same kind of thing you see with 23andMe that's using a SNP chip. And at least today it is, because it's way cheaper. But starting to do more of the genotyping for clinical purposes and doing sequencing, either with NGS, or we're starting to see more long read sequencing. There's recently a paper that I was slightly involved in called Ultra Rapid Nanopore Genome Sequencing in a Critical Care Setting. They use Deep Variant, which I have worked on for the past almost 3 years now, along with the rest of my team at Google. So we actually got to be part of like the world record ceremony for this, and it was super cool. I'm very excited to see how something like this can be done in more clinical settings around the country and around the world. Another big milestone to look out for in this third block is when we start to routinely sequence newborns, whether there's anything wrong with them or not. This is a pretty cheap investment towards a lifetime of more informed medical care, which is what you might hear referred to as precision medicine, right? You sequence the genome once, you don't have to do it again, really. And this can inform everything from what drugs will work in what doses for the person in the future, for future diseases. You can use it for disease risk prediction as well. Either more specific Mendelian diseases can be discovered at this stage, or you could just find out, oh, this person, this newborn, might have a higher risk of heart disease, and so maybe we want to make sure that they watch their diet throughout their lifetime, or get more frequent checkups for it, or things like this, right? Preventative medicine. So there's a fourth piece, which kind of goes hand in hand. It's an additional part of the third block here. Not just informing patient care, but actually making changes. So we can manipulate genotypes to change phenotypes. That sounds a little scary because we were just talking about humans a lot. So I will say this is mostly something that we do not in humans. There are many, many more places that we can do this right now. For instance, doing CRISPR and synthetic biology and so on to change plant genotypes in order to affect the phenotypes. So this is another area that is really exciting right now. Obviously, you've probably heard of CRISPR. If you're following this channel, you have probably heard of CRISPR too. And synthetic biology is another really interesting area where we can, create organisms like bacteria to accomplish certain tasks. Some exciting ones that we are hopefully getting closer to, but it's not my field, so I don't know too much of the specifics, is stuff like creating plastics that don't use fossil fuels, creating bacteria that can break down plastics. All these things could be really cool, and all would come out of synthetic biology. So like I said, I am most familiar with this base layer, and there's a lot of cool things happening in the whole rest of the stack. But whichever part you work in, you can end up having a big impact. So I'll speak for just my block here of reading the genome accurately. When we get good at that, we're unlocking more parts of the genome that we can now do GWAS on, that we can now understand the molecular mechanisms better because we can see what's going on in those genes for the first time. Whereas previously sequencing data might have been pretty confusing and you don't know which segment a certain read maps to, and so it becomes a lot harder to study anything in here. But the better we get at this foundational reading the genome accurately, it filters up to all of the other bricks, all the other layers of knowledge in this stack and layers of applications. So it is very fundamental. These two things are fundamental. Reading the genome accurately, finding connections between genotypes and phenotypes, and on top of that, we have using connections to make predictions, like informing patient care, as well as manipulating the genotypes to change phenotypes, mostly in non-humans. But there's also gene therapy and things like that, which we're getting pretty close to as well. So these are some of the trends that I'm excited about, interested to see where they go. And if you are working anywhere in the stack, I do encourage you to check out some of the efforts that are going on below you, as well as above you, because What's going on below you can filter up and inform the kinds of investigations that you will be able to unlock that might be new. If you're doing GWAS, for instance, think about, can I do GWAS better, more interestingly, by building on top of pangenomes? And similarly, if you're at the bottom here, appreciating that your work helps make these other layers somewhat more productive on unlocking new investigations for them can also help you to find interesting collaborations and so on. So one small new thing that is exciting for this channel is that today we have our first sponsor. So this video, for the first time ever, is sponsored by Brilliant. Brilliant is a creative learning platform where you can take courses in math, science, computer science, and especially great for us, computational biology. Brilliant has interactive lessons that help you build intuition and a deeper understanding of the topics. And as we've talked about before on this channel, bioinformatics sits at the intersection of biology, math and statistics, and computer science. And you can find lessons on all those three areas to dig deeper and build out your skills to make sure that you have a solid foundation in all three of these areas that join to create bioinformatics. One particular lesson that I want to highlight on Brilliant that I think is interesting. You might have seen a previous video of mine called Genes and Geography, where I ran PCA on some population genotype data. And it turns out that Brilliant has a lesson on this exact topic, but they have taken it from a slightly different angle. They have more background information. They're doing it on a different data set focused on Africa. So that's definitely worth checking out. So to get started on Brilliant for free, go to brilliant.org/omgenomics. And the first 200 listeners will also get 20% off an annual membership. Thank you, Brilliant. So I hope this video was useful to you and you are interested in checking out some of these trends. You found some interesting things to Google. So leave a comment below telling me about the trends that are going on in your area of genomics that you are most excited about. You can also subscribe to the channel. And this is a video that YouTube thinks you'll like. And this is a video that I think you'll like. I'll put something here based on this video. So thank you so much and have a great day.